Hi friends, welcome back to my channel Only Bio, presenting up the fourth part of the revision of the chapter Excretion and Osmoregulation. Friends, I hope you've been following this series of this chapter and do watch till the end because it has many important points right from the start. I mean, I have hit all the basic points plus also the important points in terms of different competitive exams. So please do watch till the end. The introduction of the excretory organs. A survey of animal kingdom presents a variety of excretory structures. In most of the invertebrates, these structures are simple tubular forms, whereas vertebrates have complex tubular organs called as kidneys. Some of these structures are mentioned here. Unicellular forms have contractile vacuoles which collect and discharge the waste products outside the cell. Friends, you may take an example of that of an amoeba, right? So contractile vacuole, as the term tells us, contractile. So this vacuole has a capacity of contraction and relaxation. So whenever it is relaxed, it starts getting filled up with all the waste products that have been collected in that organism. And as it expands to its fullest, this vacuole then moves towards the periphery, that is the side of the body or the cell, you may say, and it ruptures. Now, when it ruptures, it releases out the waste products and once again it becomes empty so that it can further collect and discharge the waste products again. So, this is how it works. It helps in both excretion as well as osmoregulation. Excretion in sponges takes place by diffusion of waste material in the water which is discharged through the osculum. You know, friends, sponges belong to porifera, right? So they have many smaller openings on the body which are called as the ostia through which the water pours in. And a single exit point is the osculum. So this is how the osculum is going to help in throwing out the waste materials that might be present in the water. True organs of excretion are found in those animals that show bilateral symmetry. Most common type is simple or branching tube that opens to the exterior through the pores called as nephridiopores. Now friends, let us understand what is meant by a nephridium. So excretory system also has got evolved. So to begin with, the first one of course is a contractile vacuole, yeah, the entire body surface that helps in the process of excretion. But an organ dedicated to this process, yeah, a cell dedicated to this process was a nephridium to start with. Okay, so nephridium is a unit of excretory system in many primitive invertebrates and also in amphioxus, and this is formed by the ingrowth of the ectoderm. This is a very important point, formed by ingrowth of the ectoderm. I can ask you this, ki nephridia are dashed by origin, so it is ectodermal by origin. Now, the nephridium, plural being nephridia, is an invertebrate organ found in pairs and performing a function sorry, similar to the vertebrate kidney. The excretory organ of many invertebrates consists of a tubule with one end opening into the body cavity and the other opening into a pore at the body surface. So on the screen you can see a tube. So one end is going to be present connected inside the body cavity, a coelomic cavity, through which the waste products are going to enter into this tubule, and ultimately this tubule will open on the exterior of the body via a small opening on the body surface that is called as a nephridiopore. It expels, that is throws, the wastes from the body cavity to the usually aquatic exterior. So in simple words, you have to remember that nephridia are normally seen in those organisms which are aquatic. The evolution of nephridia encouraged tissue specialization by eliminating the need for all the cells of an organism to be in contact with seawater for diffusion of metabolic wastes. So once again, this is hinting up at the process of evolution. So instead of the entire body surface and all the body cells involved in the same function, so this helped in what specialization process. I hope you understood this. Now friends, nephridia are of two types, Protonephridium and Metanephridium. So evolutionary point of view, if we see Protonephridium are but obvious the first which are first to be formed, they are most primitive you may say, and which later on might have got evolved to form Metanephridia. So first now we shall be discussing Protonephridium. Protonephridia. Please remember friends, nephridium is singular and nephridia is plural. So protonephridia are plural. Okay. A protonephridium, proto means first, is a network of dead end tubules. Now again to be remembered point, dead end tubules. That means one end is not going to open anywhere. It is going to open into the body cavity. Whereas the other end is going to be opening on the surface of the body cavity, that is nephridiopore. Just to give you an idea. Lacking internal openings found in the phyla Platyhelminthes, some annelids, Nemertia, Rotifera, Amphioxus, and Chordata, that is lancelets. The simpler, more primitive Protonephridia found in flatworms, ribbonworms, and rotifers are usually scattered among the other body cells. So, friends, these are all the examples of Protonephridia. Okay, I mean the phyla which can show the presence of Protonephridia. So Protonephridia is a network of dead end tubules. Okay. The protonephridium consists of a hollow cell located in the body cavity and a duct leading from it to an exterior opening called as a nephridiopore. I had told you the same. Fluid in the body cavity filters into the hollow cell located in the body cavity and a duct leading from it to an exterior opening called as an nephridiopore. The same point slightly explained in a different form so that you understand it better. Now, fluid in the body cavity filters into the hollow cell, and this can be called as a flame bulb or a flame cell, provided if it has cilia, or it can be called as a solenocyte if it has flagellum. So the cell into which this particular tubule opens up may have cilia or flagella. If it has cilia, you're going to call it as a flame cell, and if it has flagella, you're going to call it as a solenocyte. Okay, so they function in osmoregulation, that is ionoregulation. In either form, the cilia or flagellum wave the filtered urine down the tube to the outside. The terminal cells are located at the blind end of the protonephridium. Blind is nothing but the dead end, something which is closed. The perforations in the terminal cell are large enough for the small molecules to pass, but larger proteins are retained within the animal. So from this you have to understand that it is not possible to throw out the larger molecules via this. Okay, so that is the reason why there was a need felt to excrete these larger molecules. And this is how the system got evolved. Now from the bottom of the protonephridium, the solutes are led through the tube formed by the canal cells and exits the animal from a small opening formed by the nephridiopore. Selective reabsorption of useful molecules by the canal cells occurs as the solute passes down the tubule. Now, reabsorption is nothing but the absorption of substances which are important for the body so that they should not be thrown out as a waste product. Now friends, on the screen you can see the picture of that of a protonephridium. Okay, how it is, why is it called as a flame cell, how it looks like, because it has cilia. Okay, the nucleus is seen on the cap or on the anterior end. Now, Protonephridia generally found in the basal organisms such as flatworms. Protonephridia likely first arose as a way to cope with a hypotonic environment by removing excess water from the organism, that is osmoregulation. Friends, a hypotonic environment. This means it has less of solutes and more of solvents, or rather more of water. Their use as excretory and ionoregulatory structures likely arose secondarily. So these are excretory systems in the phyla Platyhelminthes and also called as blind tubules. These tubules bear a tuft of cilia or flagella. As I told you, if it has cilia, you're going to call the same as the flame cells, and if it is flagella, you're going to call it as solenocyte. An organ of excretion in flatworms. A hollow cup-shaped cell containing a bunch of cilium or flagellum whose movement draws in waste products and wafts them to the outside through a connecting tubule. That is what we can see clearly from the diagram there or the picture there. Friends, on the screen you can see the diagram that has been taken from your textbook, that is excretory system in platyhelminthes. So on the upper side you can see the example of the platyhelminthes, a flat-bodied animal, and on the sides you can see there's a network of these protonephridia seen as crossed structures, and then we can see on the lower side there's an enlarged structure given. So we have a flame cell, right, which shows a very prominent nucleus, and it opens into some ciliated structure through which a tube continues and opens to the exterior via a pore that is called as excretory pore here, but it can be also called as a nephridiopore. Hope you understand this. Now let us understand about metanephridia. Metanephridia are unbranched coiled tubes that connect to the body cavity through funnel-like structures called as nephrostomes. Friends, I know you might have got a confusion about what is meant by protonephridia and how is it different from metanephridia earlier. So in this video, in the end, we are even going to talk about the detailed differences between protonephridia and metanephridia. Okay, so hang on. Body fluid enters the nephridium through the nephrostome. So nephrostome is going to have a funnel-like structure, unlike that of protonephridia, wherein we have either cilia or flagella. So here there's a funnel-like structure, that is nephrostome, and the other end is going to open up via the nephridiopore to the outer side. And these metanephridia are seen in earthworms. The metanephridia, which occurs in the earthworm and some other annelids, open into the coelom by combining with the coelomoduct. That is an opening in the coelom you can see. The internal opening is known as nephrostome and the external opening is called as a nephridiopore. Metanephridia superficially resemble kidney tubules. A metanephridium, meta means after, is a type of excretory gland found in many types of invertebrates such as annelids, arthropods, and mollusca. In mollusca, it is known as the Bojanus organ. Friends, it's a modified structure, okay? A metanephridium typically consists of a ciliated funnel opening into the body cavity or coelom connected to a duct which may be variously glandularized, folded, or expanded, that is vesiculate, and which typically opens to the organism's exterior. Now, you may feel a few minutes back I said there are no cilia and flagella. Now, here you're saying there is a ciliated funnel. There's a difference here. Try to understand. The cell is not having cilia or flagella here. Here, the funnel, the extension might have cilia. Okay, so that's the difference. These ciliated tubules pump the water carrying surplus ions, surplus is excess, metabolic waste, toxins from the food, and useless hormones out of the organism by directing them down funnel-shaped bodies called as nephrostomes. This waste is passed out of the organism at the nephridiopore. The primary urine, that is the urine that is formed first, produced by filtration of blood or a similar functioning fluid, is modified into secondary urine through selective reabsorption by the cells lining the metanephridium. In few annelids, each segment contains a pair of metanephridia tubules bathed in coelomic fluid and encircled by capillaries. Now, friends, capillaries ought to be present there because the blood is going to consist of the metabolic waste, right? So, the metanephridia are going to collect the metabolic waste from the blood. Okay, this is why we need the capillaries there. Nephrostome collects the fluid from the coelom, that is ultrafiltration, and the first filtrate is isoosmotic, that is has the same osmotic concentration. Yeah, you can say pressure. Transport epithelia in the lumen of the tubules to reabsorb and secrete molecules. Urine exits the nephridiopore. Metanephridia are of open type and far advanced. They do not possess solenocytes. The inner preseptal, now preseptal is before the septum or the partition, and opens into coelom by a ciliated funnel called as nephrostome. Now friends, this is particularly with reference to the metanephridia that are seen in earthworms. So this is for earthworms, okay. Thus, metanephridia are open at both ends, unlike that of protonephridia, which are blind end. Okay, excretory waste diffuse from coelomic fluid or blood into the lumen of the nephridial tubule and discharged to the outside through the nephridiopore. These are found in polychaeta, that is Nenthus, oligochaeta, that is Lumbricus, and leeches. Now, friends, let us understand the major differences between protonephridia and metanephridia. First, in case of protonephridia, these are the tubular excretory structures in certain invertebrates, usually ending internally in flame cells and having an external pore. Whereas in case of metanephridia, these are the excretory organs present in many invertebrates originating in a ciliated coelomic funnel. Protonephridia are a network of dead-end tubules without internal openings, whereas metanephridia are a type of excretory glands with a ciliated funnel opening into the body cavity. Protonephridia are closed by a flame cell, whereas metanephridia have an opening to the body cavity and are not closed by flame cells. Protonephridia occur in the phyla Platyhelminthes, Nemertia, Protifera, and Chordata, that is lanceolate, whereas metanephridia occur in the phyla Annelida, Arthropoda, and Mollusca. Protonephridia occur in both coelomates and acoelomates. Metanephridia occur in coelomates. So friends, let's quickly have a recap of the types of the excretory organs. In most, and before I start with, this is going to be especially important in the MCQs wherein you can be asked which organisms or what kind of organs, yes, structures involved in excretion. This can be asked to you as a match the following as well. Yeah, I may ask you to find out the odd man out after giving you four options there. So please do focus and concentrate in this. So in most of the insects, excretion takes place by set of blind-ended tubules called as Malpighian tubules. But obviously, these are a better, what you can say, version of that of the Metanephridia. Crustaceans have green glands as excretory organs. Members of the phylum Echinodermata do not have any specialized excretory organs. The waste materials directly diffuse into the water or are excreted through the tube feet. Mammalian kidneys are a collection of functional units called as nephrons, which are well designed to extract the metabolic wastes. Friends, I hope you have been following the series. Please like, subscribe if you haven't, and hit the bell icon if you haven't yet done it. Thank you so much for being with me.